Hello everybody and welcome back to microbiology. Today we'll be going over the PCR and primer design lab. Because this course is online, we don't always get hands-on experience with these techniques. However, hopefully we'll have one benefit in that we can go over a little bit more theory when it comes to things such as cloning. To begin, we'll discuss polymerase chain reaction, abbreviated PCR. There are 4 main steps in the PCR reaction, and this is a broad overview. Afterwards, we'll go in greater detail in each one of these steps. The first step, we have our double-stranded DNA molecule represented as these 2 green lines, and the black lines represent the nucleotide base pairs that are lined up with each other and hydrogen binding to each other. The first step is called melting the DNA. And this is when the temperature is raised to a high temperature, and that causes the hydrogen bonds to break apart from one another and resulting in 2 single-stranded DNAs. Step 2 is called annealing, where what we call DNA primers have been designed to attach to each one of those single strands. These DNA primers act somewhat similarly to how primase would act in DNA replication. They basically prime the DNA strand for polymerization of your using your polymerase. That's step 3. Step 3 is elongation or polymerization, where synthesis of a new strand occurs. And then the 4th step is repeating the process. So this is cycled over and over again in order to replicate the DNA strands many, many times. The first and most commonly used polymerase that made this reaction possible is the Taq DNA Polymerase. This DNA polymerase comes from the bacterium Thermus aquaticus, and hence the name Taq is a portmanteau of the first letter of the genus and the first 2 letters of the species of this bacterium. And it was originally isolated from a hot spring in Yellowstone, and it's considered a thermophile because it thrives at 70 degrees Celsius, and we'll see why that's so important in a moment here. So the DNA polymerase from Thermus aquaticus has an optimal temperature at 72 degrees Celsius, which is pretty close to the habitat that T. aquaticus survives in. And this allows for the thermal separation of primer annealing and polymerization of new DNA molecules. In order to make this reaction successful, we need to separate those 2 processes. In the cells, these are separated by the fact that you need a primase in order to add those primers. But since we don't have a primase in this situation and we're doing it in a test tube, we use the concept of temporal and thermal separation in order to allow this to occur in 2 separate steps. PCR allows scientists to make many copies of a particular segment of DNA, but this requires a number of ingredients in order to imitate this natural process. The most obvious one is going to be the DNA template itself. Next are deoxyribonucleotides, and these will become the building blocks for synthesizing a new piece of DNA. We need not 1 but 2 different DNA primers in order to make copies of both strands of the double-stranded DNA. Of course, we need our Taq polymerase or some other sort of thermostable polymerase. And finally, a buffer. PCR allows scientists to make many copies of a particular segment of DNA. However, to imitate this natural process, there are several ingredients that must be in the PCR reaction tube. This includes, obviously, the DNA template from which the replication will occur from, deoxyribonucleotides, which are the building blocks for synthesizing the new strand of DNA, 2 DNA primers. We need 1 DNA primer that's homologous to each strand, so a total of 2 are necessary. Taq polymerase or another thermostable polymerase, and a buffer which provides the right conditions for these reactions to occur. Oftentimes, scientists will put the latter four into one mix called a master mix. This includes the deoxyribonucleotides, the two DNA primers, the Taq polymerase, and the buffer. And then that master mix can be aliquoted into several tubes, and at that point, in order to save time, the only other thing that has to be added to each tube is the DNA template. So if a medical technologist, for example, is screening different people's genes to look for a specific hereditary disease, then a master mix can be made into many tubes, and then all that's left to do is to add each patient's own template to their own test tube. Okay, so now let's get back to the process. In the first step, we said that there's a melting step, often referred to as denaturation. And to accomplish this, the sample is heated to 94 degrees Celsius, which is a little under the temperature at which water boils. So this is a very hot temperature, and as a consequence, the double-stranded DNA does not have enough strength in those hydrogen bonds between the nucleotide bases to keep it as a double strand. And what ends up happening is the double strand breaks apart into two complete single strands. So that's step one, denaturation. Step two is the annealing process. And remember that the annealing process is where primers bind to those single strands. And that is the process of annealing. And the way that this has occurred is that that sample was cooled down to roughly 58 degrees Celsius, which is just cool enough that the DNA strands can start coming back together. But with the presence of the primers there, some of these DNA strands bind with primers instead of rebinding to their own complementary DNA strand to make a double-stranded DNA. When these primers do bind to the single-stranded DNA, then we can continue on with extension. In the extension phase, we have our thermostable polymerase, which completes the job of DNA polymerase 3 in DNA replication in the cell. So these primers allow the DNA polymerase from Taq, or Thermus aquaticus, in order to bind to those primers and then begin extending or polymerizing a new strand. This requires the presence of those nucleotides, those deoxynucleotides, which are represented here as the colorful little short segments. And essentially, those come with all of the requirements necessary. They come with the ribose, the phosphate, and the nucleotide base pair in order to synthesize a new nucleotide of DNA along with the backbone. And again, this is thermally and temporally, or separated by time and temperature when this occurs. So a certain amount of time is given for each one of these steps to occur, and then we move to the next temperature. So after resting for a period of time at 56 degrees Celsius to allow the primers to anneal, then the samples are heated to 72 degrees Celsius and rested or incubated in order for the polymerase to do its job at its optimal temperature. So if we put them all together, we get the 4 steps listed here. The fourth step being the repeating process of going back to step 1. So again, in step 1, we have the melting of the DNA strands, or denaturation. Step 2, the annealing of our primers. Step 3, the polymerization step, or elongation step with our Taq polymerase. And then step 4, we're going to continue to repeat this process from the beginning over and over again, because remember, we want to get as many copies as possible. The more copies you have, the more downstream processing you can do with this DNA. And oftentimes in the microscopic world, we need an abundance of something in order to observe a signal from it. This can lead to actually quite a bit of DNA. So this is an exponential process that is kind of similar to what we've learned with binary fission with bacteria as an exponential process. This process is typically repeated on the range of 30 to 35 times. And so this is called thermal cycling because we're cycling different temperatures and we're doing so in cycles. So we go back through the process over and over again. And this leads to billions of copies of a particular DNA segment. So there's a bit of mathematics behind this. We can calculate the final theoretical yield using this formula listed here, which is basically the number of defined length sequences produced by PCR. And it is solved by using a couple variables. One is n, which is the number of cycles, and x is the number of DNA molecules in the beginning. And the formula says x times 2 to the nth power minus 2 times n plus 1. So let's do an example of this. If we have 35 PCR cycles and we only start with a single DNA, how many fragments will we have at the end of that 35 cycles? So this process isn't so difficult. We just plug in the variables into this equation. Naturally, it makes sense intuitively that we raise 2 to the number of cycles because that's what sort of defines this as being exponential. So it's 2 to the 35th power minus 2 times 35 cycles plus 1. And because we only have one strand of DNA, we have 1 at the front here. And we solve for that equation. And you can see we have a lot of DNA. So from one DNA, double strand of DNA, or one particle of DNA, we can get 34 billion strands of DNA. For an exam, you're not going to be required to memorize this equation. I will provide the equation, but you should know that n is for the number of cycles and x is the number of strands of DNA that you have. As a clue, you should recognize that n is an exponent here, and it makes sense because every time you do another cycle, you should be doubling the number of DNA molecules you have. So it's an exponential cycle, and that's why you see n as an exponent there. All right, let's go over the PCR ingredients a little bit better here. Let's cover those PCR ingredients again. The first one, the DNA template, is the source of DNA that will be used to be copied or assayed. The second is our DNA polymerase. We often use Taq polymerase for this, which is an enzyme that will copy DNA at high temperatures. In the case of Taq polymerase, 72 degrees Celsius. Then we have 2 primers, which are short sequences of nucleotides that prime the process of polymerization. And they give a starting point where Taq polymerase binds to the 3' end of the primer in order to begin its polymerization. Step 4 are the nucleotides, or what we call bases, or nucleotide bases. These are the building blocks for synthesizing new DNA strands. We use a form of it that has 3 phosphates attached, and that's because, like with adenine triphosphate, or ATP, 2 of those phosphates can be broken off, and that gives a high energy release that can be used for attaching that nucleotide base to this growing DNA strand. So this form of nucleotides we call deoxyribo because it has the ribose attached to it. and it's the deoxy form because we're talking about DNA. So deoxyribonucleotide triphosphates, abbreviated as DNTP. And finally, the buffer, which ensures a proper pH. And it includes magnesium, which forms a complex with the nucleotides so that it can be used for by the DNA polymerase. This is very important to have present. Okay, so when these cycles were first done by the original scientists, they would have a series of water baths and they would have to stand in front of them and alternate their samples between each temperature. Nowadays, because PCR is so common, we have thermal cyclers. Here's an example of a thermal cycler, and this is a programmable piece of equipment. It basically just changes the temperature of your samples through what is called a reaction block. So you can program through the software. Here's a touch screen, different times and temperatures, and this heating block will heat and cool depending on the settings that you've applied to it. And at the top is a heated inner lid also, which prevents condensation from forming on the lids of your tubes. Okay, here we have a thermal cycler programmed for its 3 different cycles. There were some previous initial steps, but the thermal cycle steps are all listed here. The first step in this was your denaturation at 94 degrees Celsius, and this occurred for 30 seconds. The second step was the primer annealing. This one was done at 57.5 degrees Celsius for another 30 seconds. And the extensions take a lot longer because you actually have an enzyme that has to, polymerize and make all of those, synthesize all of those nucleotide bases in a strand. And so this goes for 5.5 minutes. Then our step 4, it's listed as step 6 here because there were a couple extra steps that aren't visible in the screen. But our step 4 in the PCR reaction is to go back to this first step and repeat for a total of 30 times, 30x. You'll often want to have a final extension time, which kind of ties up any loose ends. So sometimes the polymerases don't finish the complete strand of DNA. And if it's important to make sure the ends are completely synthesized, you can go ahead and give an extra amount of time at your polymerization temperature in order to make sure you get full DNA strands. This particular DNA polymerase is optimally ran at 65 degrees Celsius. Most of the time though, you just see Taq being used and you'll see a program at 72 degrees Celsius. Finally, the thermocycler has this option to cool the samples down to a refrigeration temp so that Here is the first paper ever published on polymerase chain reaction. Credit for the invention of PCR was given to Kerry Mullis, and he was a very controversial, very interesting scientist who recently passed away in 2019. He was a Nobel laureate and said that he discovered PCR after an LSD trip. He was a former consultant for O.J. Simpson's legal defense team, and he created a company that infused jewelry with celebrities' DNA. So if you're interested in learning more about the guy, I highly recommend it. He has quite an interesting past. Okay, so now let's discuss primer design. In this lab, you're going to be required to design your own primers for the amplification of a particular gene. This will be kind of how your primer design will look. In this example, we have a gene, glucose 6-phosphate isomerase. This is the first enzyme used in glycolysis, and you will design a forward primer, which is the first primer, and a reverse primer, which is the second primer. The forward primer goes sort of in the direction we read, from left to right. The reverse primer goes in the direction of right to left. So I highly recommend first watching the lectures on transcription and translation before doing this lab. We're going to cover a little bit of a review from those. The first is codons. So we have DNA, which is the blueprint of life, and that's because it contains the genes that are instructions for making proteins. Copies of these genes are written into what is called messenger RNA or mRNA, and this is like a script of those genes, which can be sent to the ribosome, and then the ribosome reads that script and makes a protein out of it. Well, this script is written in a language. This language is written with words called codons, and each of these codons is a 3-letter code or 3 nucleotide bases long. These codons can code for a few different things. They can code for start codon, amino acids, or stop codons. Start and stop codons tell the ribosome where the first amino acid and the last amino acid for a protein is in the sequence. The start codon is always the same, ATG. It can also code for an amino acid, methionine, so it has a dual role there. Stop codons are TAA, TAG, and TGA, and they're highlighted in red in this table, and they just simply code for the stop codon. And you should recall that ATG would stand for adenine, thymine, and guanine. So you would see those 3 nucleotide bases in a row, 5' to 3', if that was the code given. So we can take a mRNA sequence or DNA sequence, and we can break up the code 3 by 3 for each codon, and then decipher that codon in order to see what genes it is or what amino acids they are. So here we have ATG, which is the methionine start codon. Valine would be the next one, GTG, histidine, leucine, and threonine. And if I go back here and show you again, that all comes from this codon usage table. So if we look at that first one, ATG, we would find the first letter, which is A, and then at the top of this table we see the second letter, which should be T. So we basically do, if you've ever played Battleship, we come to this both of these boxes together, and you see all 4 of these codons start with the letter A, then the letter T, and then on the far right it says third position. So the third position for ATG is the G letter. So we look at all these ATs, we find the one that ends in G, and that tells us, okay, this is methionine, or it could be a start codon if it's at the beginning of a gene. If we looked up TAA, it's right here for the letter T, second letter A, the third letter A, we see we have this red, so it's a stop codon. So you can reference a codon usage table to find each one of your codons and then decipher them. So again, all genes must have a start codon. They must have amino acids to be translated, amino acid codons, and they must have a stop codon to tell the ribosome when to stop. The start codon essentially tells the ribosome where to start reading the mRNA and writing the amino acid sequence. So a mRNA strand might include extra base pairs upstream and downstream of your gene. And so the ribosome needs to be told, okay, begin here. The stop codon does the opposite. It tells the ribosome where to stop reading and therefore stop writing more amino acids in this protein. This is a visualization of the double-stranded DNA and the sequence of amino acids it will code for. So What we're not showing here right yet is the RNA, the mRNA, but from this an mRNA script can be made. So here's the sequence, and our first start codon is ATG. It also codes for a methionine. So the ribosome is going to make a methionine first. And then our next codon is AGT, which codes for a serine. And the ribosome will continue to add amino acids to that protein until it runs into this stop codon. This particular stop codon is TGA. It tells the ribosome to stop synthesizing new amino acids in this protein strand. So recall, DNA is normally a double-stranded molecule, and each strand is anti-parallel. So what that means is we have a top strand, and we, in a cell there, it doesn't appear as it does on a screen or a piece of paper. So we have conventions in the way we write these out. The top strand is always written 5' to 3', and because they are anti-parallel, that means, here's the 5' to 3', that means that the bottom strand is going to be orientated in the opposite direction, written 3' to 5' here, in order for them to be anti-parallel. And they are also complementary. So recall that a guanine will always bind to a cytosine. And so if you were to look at every C or every G, across from it will be a G or a C, the opposite. So here we have a C, across from it is a G. And a adenine will always bind to a thymine. So again, a T will always be across from an A and vice versa. So here's our C and Gs and T and As, as you can see. They match up with each other. So in this lab, like I said, you're going to be designing your own primers. In order for PCR to work, you have to copy both strands, both the top strand and the bottom strand. So we need a forward primer and a reverse primer in order to have a successful PCR. Here I have written out a forward primer here, and then the reverse primer here for this made up gene of interest in the middle here. So don't let this overwhelm you. Primer design is a lot simpler than you might think. If you just look here carefully, you'll notice that the forward primer matches up exactly with the top strand, and so therefore it can bind to the bottom strand. So the primer that binds to the bottom strand is referred to the forward primer, and the forward primer is parallel to and matches the top strand. So see how they're both 5'? They're both going to be 5' in the beginning and 3' here at the end of the primer. And recall, this makes sense because we said that the polymerase is going to attach to the 3' end of the primer. And here we have it right here, the 3' end. The opposite is true for the reverse primer. The reverse primer is going to have the exact same sequence as the bottom strand. It will also be parallel to the bottom strand. So the 5' end and the 5' end both start with the letter C in our reverse primer and end with the letter A. And because they begin with the letter C, the C from the reverse primer can bind to the G of the top strand. Makes sense, right? Okay, so next we have something called a restriction site. A restriction site is a short sequence of DNA, and we'll cover these in greater detail in the cloning lab. But these short sequences of DNA, based upon the sequences, they can be identified by molecular scissors, which are called restriction enzymes. So it's kind of like an address for these molecular scissors. They know where to go and cut based upon the address. And each scissor has a specific address to it, which in this example, this is the NDE1 address for the NDE1 restriction enzyme. Over here we have the PST1 address for the PST1 restriction enzyme. And so in order to copy and paste the gene elsewhere, genetic engineers will add these short DNA sequences to the ends of DNA that they want to copy and paste somewhere else. So when we design our primers, these sites must be included if we want to put this DNA strand somewhere else. And in this example, we're going to be using the NDE1 and PST1 restriction sites, which we can see if the site is here, it's going to be included in our forward primer. And if the other site is here, it's going to be, that sequence is going to be included in the reverse primer that's made. So CAT-ATG is the sequence for NDE1, and then CTG-CAG is the sequence for PST1. When designing primers and restriction sites, these restriction enzymes, they need a little bit of a handle or some extra base pairs on the 5' end and 3' end in order for them to get a good grip of that sequence and make a cut. So the restrictions enzyme typically has an extra 1 to 5 base pairs added to that region. There are charts available online that shows the rate of efficiency of an enzyme, which can be dependent on how great that handle is or how many base pairs are added. These restriction enzymes are typically closer to the 5' end, and so you'll typically have to add these base pairs to the 5' end. If we just had nothing after the restriction site, usually those restriction enzymes cannot grip that restriction site and cut it. So if we look at this particular chart, I've highlighted the NDE1 site. And if we look, one base pair, it's kind of has a plus sign, 2 base pairs, a plus sign. But if we have 3 base pairs, the efficiency is 3+, which is kind of a qualitative thing saying that this is a pretty good efficiency if you had 3 extra base pairs. So we'll need to add at least 3 base pairs as a handle for NDE1. Oh, okay. One more thing, we need to make sure that this area, the 3' end, where that polymerase binds, we want to make sure it's a pretty tight binding. And CG hydrogen bonds have 3 hydrogen bonds between them, and ATs only have 2 hydrogen bonds. So GCs make really tight seals. So if we want to seal this end really well, we'll want to have 2 of the last 3 base pairs, a guanine or a cytosine nucleotide. So here we can see the last 3, we have 2 Cs. And for the reverse primer, the last 3, we have a G and a C. And now we have our melting temperature to worry about. The property of 2 strands of DNA being able to connect to each other is related to the number of hydrogen bonds or the number of base pairs in a strand. So we have 2 strands of DNA here. They're only 2 nucleotide bases long, but it kind of shows how this process works. In the top pair of nucleotides, there's a thymine that is bound to an adenine, and thymine and adenine share 2 hydrogen bonds. The bottom base pairs are a guanine and a cytosine. So guanine cytosine hydrogen bonds have a total of 3. And these hydrogen bonds act as a molecular Velcro that allows these 2 DNA strands to stick together. The more hydrogen bonding, the better they stick to each other. The more Velcro hooks that are there to attach one strand to the next. So in order to have a primer that sticks well enough, we need to have enough of these hydrogen bonds there. And so that's how we determine the length of our primers. So for this, you'll have to remember that AT bonds have 2 hydrogen bonds, and GC bonds have 3. And so we're going to use the melting temperature to calculate how many base pairs we need in our primer. This is because the temperature at which a primer will anneal is related to the number of these nucleotides that it's homologous to. The more nucleotides and the more hydrogen bonds, the higher the temperature at which these primers are going to anneal. So what is melting temperature specifically? The melting temperature, abbreviated as Tm, is the temperature at which 50% of the primers in a solution are annealed to the DNA and 50% are melted. Recall that the warmer the solution, the more melted the DNA becomes. Remember, our melting temperature is really hot. It's 94 degrees Celsius. As you cool, the DNA strands begin to slow down in their movement around in the solution, and they slow down enough that the hydrogen bonds can grab 2 single strands of DNA and attach them together. The Tm can be roughly calculated based upon the number of hydrogen bonds. If we took the sum of the AT bonds and the GC bonds, we can calculate our Tm. So AT bonds, they only have 2 hydrogen bonds. They're not very strong. They only count as 2 degrees Celsius. GC bonds count as 4 degrees Celsius. So if we had a Ford primer that had this sequence to it, ATC ... ends in ACC, if we count up all of the A's and T's in this sequence, we find that we have 13. If we count up all the C's and G's, we find that we have 9. And remember, we're going to give 2 degrees Celsius per AT bond and 4 degrees Celsius for every GC. So 14 times 2 degrees Celsius plus 9 times 4 degrees Celsius is going to equal 28 degrees Celsius plus 36 degrees Celsius, giving us a Tm of 64 degrees Tm. So at 64 degrees Celsius, 50% of our primers will be attached and 50% of our primers will not be attached to a DNA strand. As we cool this more and more, that's going to allow more DNA primer complexes to form. The hotter it gets than 64, the fewer primers will be attached to their DNA strands. So 50% attached and 50% not attached is not a very good ratio. Again, we want to get as many copies of our DNA as possible made. So we want more than 50% of our primers attached. So what we're going to use is our TA. That is our actual annealing temperature that we program into our thermocycler. A TA that is too high causes annealing to be insufficient. So this would lead to low PCR yields. A TA that is too low leads to primers that can actually attach in other regions of the DNA template. This is referred to as non-specific binding. So we want to avoid either of these, and so the sweet spot is typically 5 to 10 degrees Celsius less than the Tm that was calculated. So in this previous slide, we calculated the Tm of 64. We would want to run this somewhere then between 54 and 59 degrees Celsius. So there's several documents that you'll use for this assignment. The first one is an instructions document, which I will have posted. It will be the PCR and Primer Design Lab instructions. The next one is a document for downloading Snapgene. Snapgene is a software you'll use for your primer design, and we'll have instructions on how to download and use the free trial version of that. Next will be the green fluorescent protein sequence. This is the actual gene you're going to be cloning for this lab and the next lab. And Lastly, when you're all done, you'll take the PCR primer design Canvas quiz. This will just be like a lot of these other labs, a series of questions that you'll have to answer. And then you'll get your credit for the assignment through this quiz. Anyway, I really hope you all enjoy this, this lab. It will be a two-part lab, and the second part will be the cloning lab for next week.